You start out as just one cell, a fertilized egg. It's made when an egg cell from your mother fuses with a sperm cell from your father. How do we get to have trillions of cells from just one cell? The answer is mitosis, otherwise known as cell division. Cells have the capacity to make more cells by dividing. Mitosis generally results in two daughter cells that are identical to the original parent cell. You, the fertilized egg, first divided into two cells. Those two cells became 4, then 8, then 16, and so on until you became a fully functioning human being. Pretty cool, hey? Mitosis is a very organized and strictly regulated process when genetic information stored in the original parent cell is perfectly copied and transmitted to each daughter cell. Mitosis has 4 stages. These stages are called prophase, metaphase, anaphase, and telophase. Cytokinesis occurs after the 4 stages of mitosis and involves the splitting of the cells into 2 daughter cells. Between cell divisions, there is a stage called interphase. Let's quickly talk about each of these stages. Most cells in your body spend the majority of their time in interphase. This is when your cells carry out their normal daily cellular activities, such as obtaining nutrients, growing, and making molecules that the cell needs to survive and carry out its specialized function. In interphase, cells also undergo DNA replication in preparation for cell division. DNA replication is when cells make an identical copy of their DNA that will be passed on to one of the daughter cells during mitosis. We will go over the basic concept of DNA replication, but first, let's look at the structure of DNA. DNA looks like a twisted ladder. The rungs are made up of 4 different kinds of bases: cytosine, thymine, adenine, and guanine. Cytosine always pairs with guanine, and adenine always pairs with thymine. The sides of the ladder are known as the backbone of DNA, and it's made up of repeating units of phosphate molecules and deoxyribose sugars. When DNA is unzipped down its middle, each strand forms a template for the other side. After complementary nucleotide bases are added, we are left with 2 identical double-stranded DNA molecules. This is how DNA is copied in your cells so that a complete set of DNA is passed down to each of the new daughter cells during mitosis. During prophase, the nuclear envelope breaks down and the DNA of your chromosomes condenses into structures that look like X's. Each strand on the X is called a chromatid, and each of these chromatids are identical. During cell division, one cell will receive one chromatid and the second cell will receive the other. In metaphase, the chromosomes are arranged in a line through the middle of the cell, called the metaphase plate. During anaphase, the chromosomes split and move to opposite sides of the cell. Each side of the cell receives one complete set of DNA. In telophase, the nuclear envelopes begin to form around each set of DNA, and the chromosomes unwind into their less condensed form. Cytokinesis is not considered a stage of mitosis, but it's necessary to complete cell division. It involves the pinching of the cell membrane between the 2 separately forming nuclei until the cells detach from one another. The daughter cells are then free to grow and live as separate cells.